Hello, my dear children. Namaste and welcome to yet another session of our ICSE series for class 10. Many of you have been asking me, ma'am, can you do a CBSE class 9 session on this topic? But again, I think I told you this in my previous session also. Children, this channel is meant for all of you. The only difference in this series, our newly launched series, is that we are going by the order, or at least the chapters which are in the ICSE children's syllabus. But that doesn't mean that if you are a CBSE or a state board or a 9th grade or 11th or 12th grade student, you cannot attend or watch any of the sessions. You can definitely attend it as long as the topic is relevant to you. So my sincere suggestion is, please do subscribe to the channel. Please do have a look at the daily schedule that's put up. Look at what topic is going to be done in today's session, and if it is relevant to you, if you would like to just know more about it, or if it's relevant to your syllabus too, definitely my suggestion would be to attend it. The more you hear about a concept, the better it gets retained in your memory and you understand it better, right? You get to, because different teachers teach in different styles, so you may get some new insights from attending any of the session as long as you are interested in knowing about the topic. Okay, so let's not waste any more time. You know that we have already started with the chapter, and we are done with the chapter cell cycle and cell division. We spoke about cell cycle in general. We spoke about mitosis, meiosis, and in our last class we have also discussed about a few very important terms, which are chromosomes, the structure of chromosomes, the basic structure of the chromosome, what is chromatin, and what are chromatids. And I hope chromatin, chromatids and chromosomes are very clear to you in your head now. So today it is time to get started with our next topic, which is the structure of DNA. Again, it's not a very complex topic, but a very, very important one, just so that you would understand some of your future chapters, especially like genetics and other chapters, a little better. And if you are going to be continuing biology in your 11th and 12th standard, this will certainly be of great use to understand this as your base right now. Okay, so don't go anywhere. Stay tuned. This is going to be a simple session. Make sure you understand the concept very well. Okay, but before we start, this is the homework question that we had in yesterday's session. How many autosomes and sex chromosomes would be found in a human sperm cell? I did get a lot of very quick answers, and what I was surprised about was that most of you gave the answer as 23, which is not wrong, but not the most appropriate answer, because my question here specifically says how many autosomes and sex chromosomes. Autosomes and sex chromosomes have been specified. So I'm asking you about the specific number of these two types. Remember, in the previous session, we discussed that chromosomes are of two types, autosomes and sex chromosomes, right? And we have also seen what they are. Those of you who have understood, great, you have done a great job. The rest of you, perhaps you did not notice that I'm asking these two differently or specifically, which is perhaps why you gave the answer as 23. Now, 23 would be the right or the most appropriate answer if I had asked you how many chromosomes would be found in a human sperm cell. How many chromosomes in general? Now that I have specified, the answer would be a little different, and I saw that the answer was given only by one person completely correctly. Let's look at who that was. Now, these are the answers, or the quickest answers that I got, and these are from the chats that came up while the session was being premiered. The first answer I got from Vikash Singh, I think 23. Well, Vikash, it's not wrong, like I said, but it's not the most appropriate, because I'm asking a more specific question. Again, Krishna Bhaskar, you have also said the answer is 23, and I have the same reply to tell you. And the correct answer was given by Yogesh Raja, 22 plus X or 22 plus Y. Great job, Yogesh. That shows that you have understood what the concept is completely and what the question was completely. But those of you who answered it as 23, please pay attention to me for the next 2 minutes. We will just have a quick recap of that so that this is much clearer in your head, right? In human beings, there are, in a typical human diploid cell, how would you represent a diploid number? You would represent it as 2n, right? The 2n number in a typical human cell is 46, correct? And of these 46 chromosomes, there are 2 different types of chromosomes, okay? They are autosomes and sex chromosomes. Now, how many of each of these are present? I've already told you yesterday, but just revising it again, recalling it again, out of 46, 44 are autosomes, while the remaining 2 would be sex chromosomes, which in the case of a human female would be 2 X chromosomes, while in the case of a male diploid cell would have 44 autosomes and an X and a Y chromosome. Okay, this is extremely important to understand. Have this clear in your head. This is the case with the human female and this is the case with the human male. And I'm talking about the diploid cells of our body. Okay, so what are sex chromosomes again? Sex chromosomes are those which determine your gender or your sex, whether you're a male or a female. Okay, if the zygotic cell has 2 X chromosomes, that ends up becoming a girl baby, a baby girl or a female baby. If the zygotic cell has an X and a Y chromosome, that ends up developing into a baby boy or a male baby. Okay, have this very clear in your head. So what are autosomes? All the other chromosomes in your cell which do not determine your sex or your gender are called autosomes, and they carry the genes for the various other characters of your body. Okay, so this is about a typical diploid cell. But remember, we also learned about what haploid cells are, right? In the human body, only the sex cells or the gametes are the haploid cells. Haploid would be half of diploid. Right? So what would the autosomal and sex chromosome number in a gamete? This was exactly my question to you yesterday, right? If it is an egg cell, okay, an egg cell or an ovum, the gamete of a female, it would have 22 autosomes and an X chromosome, which is exactly half of this. 22 is a half of 44. Instead of 2 X chromosomes, I have 1 X chromosome. Whereas in the case of a male, what happens is sperms can be of 2 different types. 50% of the sperms, approximately 50% of the sperms produced by a male would have 22 autosomes and an X chromosome, while the remaining sperms, the remaining 50% of the sperms would have 22 autosomes and a Y chromosome. Okay, which is why I was telling you that Yogesh's answer here is the most appropriate and the most correct one, 22 plus X or 22 plus Y. Thank you very much once again, Yogesh, for coming up with a quick and correct answer. Now you can relate it to the question, how many autosomes and sex chromosomes would be found in a human sperm cell? If I had asked you in a human egg cell, your answer should have been 22 plus X, 22 autosomes and 1 X chromosome. Since the question was sperm, this would be the correct answer. I hope you have got this completely clear in your head. It's a very important concept to understand. I keep repeating this because our next unit or the next chapter will be genetics, all about heredity. This concept is extremely important. We may not have the time to sit and again recall all of these while we are doing that chapter, right? So have this clear in your head before we start with the session on Monday, before we start with the next chapter on Monday. Okay, so going ahead, This is again, I thought, a very important thing to tell you. Don't be pushed around by fears in your mind. Be led by the dreams in your heart. That's an amazing saying, right? Because as human beings, all of us have a lot of fears. We have a lot of insecurity for different reasons. For each of us, it may be different. But at the same time, all of us have a lot of dreams. We have a lot of ambitions. At the moment, I assume that your dream is to perform really well in your class 10 board exam. So children, my suggestion to you, my advice to you is that focus only on that. Forget about all your fears. Get rid of all your negative thoughts. Don't allow yourself to be pushed by any of your negative thoughts. Just be concerned about your dreams. That is all that matters. Have a deep desire that you will win. We are also here to support you and we will together reach success. Okay? So let's get started. So before we continue because today's session will be a continuation of our previous session, I thought it would be nice to have a quick recap, which today is going to be in the form of a quiz. Okay, now this is not a live session, as you know, but I will be looking at each of your chats. I will be watching how many of you are giving the correct answer and how quickly, as and when I'm asking you the quiz question. Okay, so the very first question is this. Duplicated chromosomes are joined at a point termed centriole, centromere, centrosome, and chromatid. Be very quick with your answers, children. 3, 2, 1, go. And the correct answer is centromere, right? Duplicated chromosomes, I mean sister chromatids, are joined at the point that we call centromere, right? That is the correct answer. Coming to the next question, the cell component visible only during cell division. Chromosome, chromatin, mitochondria, and cell membrane. Again, be very, very quick with your answers, children. 5, 4, 3, 2, 1, go. And the correct answer is chromosome. Right? During cell division is only when you would see the structure in the form of chromosome. Until then, in the interface, you would find it only as a thread-like structure, which is called chromatin. Right? So, coming to the next question, chromosomes that do not determine the sex of an individual are called. You must be extremely quick in answering this because we started today's session recalling this particular concept also. So, be very, very quick. 3, 2, 1, go. The answer is autosomes. I've told you this. And those chromosomes that determine the sex of an individual are called sex chromosomes. Okay? Now, identify the type of chromosome in this picture. Remember, yesterday we learned that there are 4 different types of chromosomes based on the position of the centromere, which is why I have put those as options here. Look at the picture and tell me what type of chromosome it is. 5, 4, 3, 2, 1, go. The answer is acrocentric. And what exactly is acrocentric? Acrocentric chromosome is the type of chromosome in which the centromere is slightly away from the tip of the chromosome. If it is at the tip, it would be telocentric. If it's at the middle, it's called metacentric. If it's slightly away from the middle, it's called submetacentric. So I hope that is completely clear. Great job, guys. Keep going. Let us now continue and start with the next one, the topic for today. So this is where we stopped yesterday. You must be able to recall this. This is the chromatin that you see here. Remember, we compare it with a bowl of noodles. It's just the nucleus, which is the bowl here. The chromatin, which is looking like the noodles here. And when you zoom into it, you would see that chromatin is nothing but tightly packed DNA. And you see the DNA here. It's arranged in the form of histones, nucleosomes. We discussed all of these terms in our previous classes. And this is what you would see in the most zoomed in part of a chromatin, or completely unwound chromatin. You would see the helical form of DNA. Now that again is an amazing structure that was discovered by two scientists, James Watson and Francis Crick. Okay, when did this happen? This happened somewhere around the early 1950s, somewhere around 1953, 54 around, when these two scientists were together working in a laboratory in England. They did a lot of things, like after a lot of research, after years of research, they discovered the structure of DNA that we know today and we call today as the double helical structure, or the double helix of DNA. Okay, so if you look at it, one DNA structure, now I'm sure when I tell you DNA, it's perhaps a double helical structure that comes to your mind, right? Something like this, right? So it's important to understand what goes into it to give it that particular structure. That's what we are looking at here. Now, the first and foremost thing to remember is that the expansion of DNA is, yes, tell me? Yes, deoxyribonucleic acid. Deoxyribonucleic acid. This is what is short formed or abbreviated as DNA. We keep saying DNA, 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 but what is it? Nucleic acids are basically polymers. Right? You know what polymers are. They are made up of repeating units of monomers, or smaller subunits. Right? So nucleic acids are made up of long chains of subunits which are called nucleotides. Okay? And what exactly is one nucleotide? One nucleotide consists of three parts, which are a sugar, a phosphate, and 4 nitrogenous bases. Remember that much for now. A nucleotide is made up of a sugar, phosphate, and 1 nitrogenous base. Okay, we have 4 options, out of which one would be part of a nucleotide. Okay, this is roughly the structure of a nucleotide. Okay, a sugar, a phosphate group, and a nitrogenous base. Look at that, S, P, B. Now, one of my favorite Indian singers is Shri S.P. Balasubramaniam. So, I'm not sure how many of you know him, but he's one of the legendary singers of India, like sung mostly in Tamil, Telugu, a few in Malayalam, a few in Hindi also, some of the evergreen songs. One of the senior most artists in the film and music industry. So for me, it's easy to remember with SPB. S.P. Balasubramaniam, normally people call him SPB sir. So SPB, sugar, phosphate, base. Okay, it's easy to remember it like that. What is the sugar? What form of sugar? It's a type of sugar which is called deoxyribose. Okay, a deoxyribose sugar, then a phosphate group, and what are these nitrogenous bases that I am talking about? They are adenine, guanine, cytosine, and thymine. Okay, adenine, guanine, cytosine, and thymine. Now, adenine and guanine are together called purines. Okay, and cytosine and thymine are together called pyrimidines. So if I ask you, give me an example of a purine, you have to tell me either adenine or guanine. And if I ask you pyrimidine, you have to tell me either cytosine or thymine. Now again, it's easier to remember if you just think of it as A, G, C, and T. Okay, this is how it is. They are always represented when you represent the structure of a DNA. They are always written in alphabets, only the first alphabets, A, G, C, T. These are in fact the 4 alphabets or the 4 letters which make up the language of life. Okay, adenine, guanine, cytosine, thymine. Very, very important, children. Remember, these two are purines and these two are pyrimidines. That's just something to do with the classification, the biochemical classification of these molecules. The biochemical category into which these two fall are called purines, and these two fall into the category called pyrimidines. Okay, and get the spellings right also, because they are very, very important. One common mistake that children generally make, even teachers do make that mistake often, thymine. Okay, the spelling of thymine is T-H-Y-M-I-N-E. It's not T-H-I-A-M-I-N-E. So keep that in mind. The T-H-I-A-M-I-N-E is a vitamin. T-H-Y-M-I-N-E is the nitrogenous base that we are talking about today. Okay, so look at the structure. This is one single nucleotide which has a deoxyribose sugar, a phosphate group attached to it, and a nitrogenous base. Why I have written one of these four is this could be either one of these four. It would only be one of these attached to one single nucleotide. Okay, and that is only a nucleotide. A chain of many such nucleotides would together form a nucleic acid, a deoxyribonucleic acid, or DNA. Okay, now let us look at a slightly more complex structure of the polymer as a whole, the structure of the DNA. Now in this image you can see that the backbone of the DNA structure is made up of sugar and phosphate. Phosphate, sugar, alternating. You can see it attached to each other and forming the backbone. And within that, you can see the base pairs. Now here, it's not just any random alphabets that are attached to each other. And why do I have two rows? That is very important to understand. This is because DNA is a double helical structure. It's made up of two strands, okay, running parallel to each other. This is one of the strands. This is the strand that is parallel to it. Okay, so sugar, phosphate, and a nitrogen base. Here also you have a sugar, phosphate, and a nitrogen base. Now, the most important part comes in here. The two bases bond with each other using hydrogen bonds, and that is how they form these base pairs. One nitrogen base bonds with another nitrogen base to form a base pair. You can see that in the form of steps here, right? In every row, they are forming something like the steps of a ladder. You can compare it to a ladder, at least in this form. Imagine that these two are the sides of the ladder and these are the steps or the rungs of a ladder. Okay? And there is a rule for this pairing. It's not just any random thing. It's always adenine that pairs with thymine. A and T always are together, that too with a double bond, a double hydrogen bond. And guanine always pairs with cytosine through a triple hydrogen bond. A and C pair with each other. Sorry, A and T, once again, A and T pair with each other, G and C pair with each other. Okay, very, very important, children. Just keep in mind that one purine always pairs with another pyrimidine. Two purines or two pyrimidines cannot bond with each other. The one base pair should always be composed of one purine and one pyrimidine. As you can see, A and T here, G and C here, right? See, this is a double bond here, A and T. This is a triple bond here between G and C. G3C, remember that it's rhyming, right? To say it like that, G3C, forming a bond here. Again, here it's TA. The order doesn't matter, but who they hold hands with matters a lot. T and A or A and T, C and G or G and C. Right? That is exactly what I have written here for you. In a DNA molecule, purines and pyrimidines are always equal in number. And this is because a purine always pairs with a pyrimidine. So if there is a purine, if there is an A somewhere in a DNA helix, it means that it is always bonded with the corresponding T in the opposite strand. right? Keep this in mind. It's a very, very important rule. Now, sugar and phosphate, we have seen this, form the backbone of the DNA molecule. You are clear with that by now, right? I suppose this is very clear to you. Now, let us look at the next image, in which I have compared the image that we have understood now with the DNA image that you are generally familiar with. Now, I told you that you can compare this to a ladder. Now, imagine that I'm just twisting it. I'm holding the two ends of the ladder and just twisting it. It becomes something like this. More than a twisted ladder, it's in fact more appropriate if you compare this structure to a spiral staircase. All of you must have seen a spiral staircase, right? Somewhere or the other. You can compare this with that. That is exactly what a DNA double helix looks like. Here again, you can see adenine, thymine, guanine, cytosine. You can see the color coding also that way, and the blue strands here are your sugar phosphate backbone. Just compare these two. Just take 4-5 seconds, compare these two and get it clear in your head. Okay, children? Now, let us relate what we have studied today to what we have done in the previous class. We started with a cell. We have the nucleus. If you zoom into the nucleus, there is chromatin there. And if you pull out a thread of chromatin, you would find chromosomal structures, especially if it's in prophase or a cell division phase. This is what you would see it like. But if you pull it out and stretch it further, the DNA, the long thread of DNA would come out, and you would see that it's arranged around the histone proteins, and DNA and histones wound around each other. DNA wound around histones, and forming a complex would form a structure called nucleosomes, right? And further, nucleosomes undergo coiling to form the structure that we see as chromatin, right? The long structure chromatin that has been tightly and intelligently and smartly packed into the nucleus of the cell. Okay? That is what we have done in the previous class. And now, if you just zoom into the DNA structure, you can relate it to what we have done today, what we have discussed today. The nucleotide base pairs, right? Guanine, cytosine, adenine, and thymine. The color codings are here. You see that A, T are always together. G, C are always together. Again, remember that A and G are the purines and T and C are the pyrimidines. I'll tell you one more small trick to remember that, if that is confusing again. A, G are purines and T, C are pyrimidines. Okay? What do you relate to AG? Think of my name, Ambika Gopalakrishnan. AG would be my short form. My first name and my second name put into a short form together would be purines. And T and C, maybe TC, the TC that you get from your school or something like that, think of something like that, would be pyrimidines. And one purine always pairs with another pyrimidine. And it is always AT and GC. Here, how do you remember this? G, C. G and C are rhyming and they look alike. G and C therefore prefer to go together in a base pair using a triple hydrogen bond, while the remaining ones, A and T, bond by a double hydrogen bond, right? Okay, now that you have understood what DNA is, what chromosomes are, most of us have a question in our minds. What is DNA and what is gene? Aren't they one and the same? Well, the answer is that it's partly correct. It's pretty much the same because DNA is what contains all your genetic information. But specifically speaking, one gene is a single unit of heredity. In fact, the easiest way to explain it to you would be, now that you know the structure of DNA, one segment of DNA, a small segment of DNA, is called a gene. Okay? That is why I have marked this like this in this image. Now let us look at another image wherein I have put the chromosome, DNA and genes. Okay, we have the chromosome, which we all know about. If you just pull it out, you would have a thread of DNA, and in that, one section of it is one gene, next section of it is another gene, and so on. We have so many different genes because each of them has a different responsibility of deciding what our respective characters would look like. For each and every character that we have, hair color, eye color, height, everything, right? Everything and anything related to us are decided by the genes, and genes are segments of DNA. And in every cell of your body, you would have genes in this form. You would have the DNA and segments. Those segments would remain the same in every single cell, right? So DNA and gene. Understand the difference, understand the relationship. This is very, very important. Now, what do I have here? Now, this is gene 2 that's been zoomed out and put in here. Imagine that. Now, I told you we write it in letters, right? A, T, G, C are just the 4 different letters that we use. Okay, so, and keep in mind the base pairing rules. I told you A always pairs with T, G always pairs with C. Keeping that in mind, see if this is making sense to you. This strand of DNA is GT. CAGATCC ATTGAC, and the corresponding strand, okay, has its complementary. G is always pairing with C, and that way you should be able to write what the sequence of the opposite strand would be. Perhaps this is not very important as far as your 10th standard board exams are concerned, but if you practice a lot of these base pairings and predicting the sequence of the opposite strand, if you are given this particular strand, I'm sure you will never forget the rules of DNA base pairing. Okay, so one more thing, just to recap what we have done. We know that the DNA is a long nucleic acid. It's a long polymer made up of repeating units of nucleotides. Each nucleotide is made up of a deoxyribose sugar, a phosphate group, and a nitrogenous base, which can be one among A, T, G, C. Okay, so imagine the magic, right? A, T, G, C. It's just 4 letters that you can really say it forms the language of life. Different permutations and combinations, different combinations of just these 4 letters are what form the different genes in our body, and those differences are what make us unique. In fact, it is said that these alphabetic combinations in every individual, in every person's DNA, it's said that about 99.8 or 99.9% of human beings have exactly the same alphabetic order of DNA, same basis in the same sequence in their genes. It's only the 0.1% difference that makes us who we are. It's actually amazing because you can compare it to the alphabets, the 26 alphabets that we have in the English language. Together they make up so many different words. Using so many different words, we make up so many sentences. Similarly, in music, we have 7 different notes. It's always do, re, mi, fa, sol, la, ti in English, or sa, re, ga, ma, pa, da, ni in Indian music. So just different combinations of that make so many different types of music, and in English so many different types of words. Just like that, just a combination of 4 letters A, T, G, C make up what we are today and decide everything about us. Okay, so that is how important these nitrogen bases are. One more thing I'd like to add here. Now, remember I told you it was James Watson and Francis Crick who discovered the double helical structure of the DNA, and they also won the Nobel Prize for this. But there is one interesting story that some people are not aware of. In fact, when these 2 scientists were working in their lab, there was another scientist by the name Rosalind Franklin who was working in another lab in England. So she was also doing like similar experiments, and she was an expert expert in taking X-ray crystallography. Okay, X-ray crystallography was her specialization. So she had taken a lot of amazing pictures. She and one of her students had taken some amazing images of the DNA structure, which unfortunately one of her rivals by the name Morris Wilkins, who worked in the same lab as her but was friends with Watson and Crick. He in fact showed those pictures to Watson and Crick while they were doing their own experiments. And once Watson and Crick saw one of the pictures that Rosalind Franklin had taken during her X-ray crystallography imaging techniques, they in fact, like, Franklin was not at all aware of this. Okay, secretly it reached them. They saw this, and it was after being inspired by that they were able to build the double helical model of DNA that we know today. Unfortunately, Rosalind Franklin also passed away at a very young age because of ovarian cancer, and it was only these two scientists who shared the Nobel Prize for that. And only much later, Watson and Crick even revealed to the world that it was Rosalind Franklin's image 51. It's called photo 51. It was only because of Rosalind Franklin's photo 51 that we were able to build up and arrive at the model of DNA that we have discovered today. So as a woman also, and as a human being also, I always think in my class, it's very important to give equal credits to Rosalind Franklin also. She was also a great scientist. So obviously, let's give credits to all three of them for the discovery of the double helical structure of DNA. But of course, if it's asked for your exam, you can just write Watson and Crick, James Watson and Francis Crick. Okay, so now let us have a quick quiz time before winding up. Gosh, so many different terms. This will be a very quick quiz, just about 2 or 3 questions that I have put based on what we have learned today. The first question is, adenine pairs with cytosine, guanine, thymine, or A and B. Come on, be very quick. Adenine pairs with. I have told you all the base pairing rules. Based on that, you should be able to tell me the answer right now. Okay, and your time is up. The answer is thymine, AT. Remember that, AT always pair together. The next one. Which of these make up a nucleotide? Deoxyribose sugar, phosphate, nitrogen base, ribose sugar. Now, I must tell you, more than one answer is correct for this. It can be one option that's correct. It can be 2, 3, or 4. Whatever you think the correct options are, it's a multi-select question. Put in your answers right now. 5, 4, 3, 2, 1, and go. The answer is A, B, and C. One nucleotide is made up of deoxyribose sugar, phosphate, and a nitrogen base. Remember the simple rule SPB. Those of you who know SPB sir as a singer can definitely relate to him. If you don't, you still can make a mnemonic or you can relate to him. So keep that in mind so that you will never forget this. SPB together makes a nucleotide. Many nucleotides together make a nucleic acid, which we know as DNA. What about ribose sugar? Why is that not the answer? Now, deoxyribose sugar and ribose sugar are two different types of sugars. Okay, deoxyribose sugar is present in DNA. Ribose sugar is in fact the type of sugar present in RNA, which is another type of genetic material. As of now, we are not concerned about that. DNA is all that we need to understand. And here is your homework question for the day. If the DNA sequence on one strand reads ACG, TTCG, AATC, then the sequence on the complementary strand would be dash. Look at the question very clearly. Come up with the quickest possible answer. Think of the base pairing rules. Let's see who is giving the fastest and most correct answer. Again, this cannot have multiple answers because it is just crystal clear if you know the rules. Okay, so I think we are done for the day. Our next sessions will be related to the chapter genetics or heredity and evolution. Heredity, we'll start off with the basics of heredity. So I'll see you in a very, very interesting session in our next class. But before that, in the meantime, remember that the secret to getting ahead is always getting started. You know that I have given you multiple tips and tricks about how to make a timetable, how to score 100% in ICSE boards. All of those things, I've made enough motivational videos for you, and we will continue to make them in the days to come. Also, just in case you have not started your preps with a systematic timetable, do get started right now. The secret to getting ahead is always getting started right. Start with full motivation, go ahead with full josh. We are always here to help you. We are in this journey together. And do not forget to download Vedantu's learning app, and do remember to join our Telegram group. And most importantly, if you have liked this session, if you have liked my previous session, do not forget to hit the like button right now. If you think any of your friends of different boards, the same board, different grades or same grade or whatever, if you think anybody would benefit from knowing this, do not forget to hit the share button and share it with all your near and dear ones. And do remember to subscribe to our channel Vedantu 9th and 10th, because we are continuing to work hard to give you a lot of amazing sessions even in the days and weeks to come. You can also connect with me at ambika.gopalakrishnan@vedantu.com, or if you have any comments or any questions regarding anything that I can help you with, or any of our sessions that we have done so far, you can even post it in the comment section below. And until I see you again in a much more livelier session, happy studying and bye-bye.